This is TWIS. This Week in Science, episode number 860, recorded on Wednesday, January 26th, 2022. How to regenerate a leg. I know you want to know. I'm Dr. Kiki, and today on the show, we will fill your heads with frog legs, flirtation, and friends. But first, thanks to our amazing Patreon sponsors for their generous support of TWIS. You can become a part of the Patreon community at patreon.com/thisweekinscience. Disclaimer, disclaimer, disclaimer. You need to start somewhere. An inspiration, a twinkle in an eye. It doesn't come from nowhere. Sometimes it's a good book, a walk in a park, a nice long shower. Sometimes it's getting the sleep you need. It's drawing outside the lines or staying inside a box. Sometimes it's a bunch of cells struck by electricity in a muddy cocoon. Or ingredients stirred in a pot at just the right temperature. Or it could be This Week in Science. Coming up next.
I've got the kind of mind that can't get enough. I wanna learn everything. I wanna fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I wanna know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Good science to you, Kiki.
And a good science to you too, Blair. And welcome everyone to another episode of This Week in Science. Everyone out there, we're so glad that you're joining us once again for another fun-filled adventure through the world of science. Woo-woo! But you might have noticed already that we're missing a Justin. So there's news. There's news! Justin's not joining us tonight because he's on baby duty. So congratulations to Justin and Yana for their newborn child. There's a new baby in the TWIS family.
Life hath sprung.
A little life just flickering right there. So exciting. So there's not much sleep being had in Justin's world right now. But we will keep the science fires burning and are really looking forward to seeing when he comes back. But for now, we'll let him be off being a daddy.
Yeah.
Papa Justin. But in the meantime, there's still the science. We're going to keep it going, right, Blair?
You betcha. Yeah, I was gonna say you got it, and then you betcha, and then you bet it was almost what I said.
you bet something. Don't bet on betting's bad. Okay.
You got it, Kiki.
On this show of science tonight, we have all sorts of science news. I have stories about sweet tasting, deep microbes, and frog legs. So many good things. What is in the animal corner?
Oh, well, outside of the animal corner, I brought a bunch of dinosaur stories, still, animal stuff, but we'll talk about that first. And then I have hippos and jaguars in the actual animal corner proper.
I like the hippos and I like the jaguars, and they're both in the same part of the alphabet. H-I-J, it's right in there. Yes.
Some nice megafauna for the animal corner.
Yes. But now it's time for us to start the show. So as we jump into the show, I want everyone to remember that if you are not yet subscribed, you can find us all places that podcasts are found. We also stream live every week, Wednesday at 8 p.m. Pacific time on Facebook, YouTube, and Twitch. We are TWIS Science on Twitch and Twitter and Instagram, and we are This Week in Science everywhere else. You can find information about the show and show notes and all those things at twis.org. Okay, let's start with the science. Big news this week. The naysayers, I think I might have been concerned and counted as one of them, have been so far proven wrong the James Webb Space Telescope has made its way to Lagrange point 2 in orbit around Lagrange point 2. It just had a booster exercise that put it into its orbit, and now with all of its solar shades and mirrors unfurled, it is now ready to start the 5-month-long process of calibration and testing to make sure it's ready to actually do observational science and to get on with its mission. So it's gotten where it needs to be. It's doing what it needs to do. It has very successfully, like more than successfully, it has done better than expected all the way out. So now, because of the way that they launched it and certain things, they're going to be able to have a longer lifetime to it because they have more fuel than they thought they would. So there are all sorts of things going on out there. And maybe we will have a wonderful infrared view of our universe by June.
Yeah, whatever the opposite of it breaking is what happened.
Right?
That's great. I have never been so happy to have a prediction already foiled. Hopefully it stays that way.
Yeah. So, I mean, there's 5 months still of the calibration and the testing and making sure all the systems work and there's a lot that can still go wrong. But it's gotten where it needs to be. And that is the big part. I mean, I think some of the interesting questions, there are other the Deep Space Observatory is out around L2. And it's a big question. My son was asking me this last week. He's like, what if something does go wrong out there? How do they fix it? And I was like, well, they don't. And he's like, why don't we have a space station out there? I'm like, yeah, but these are great questions. It's not something that'll be done right away, but I mean, they're very interesting questions to think
about. You need
about.
to send a shuttle out there with a 3D printer that will print a robot to then do the actual repair. Yeah. So it's not sitting out there also getting ruined, but instead you're like, oh, let me send the shuttle that is only as large as a 3D printer. There you go.
One day we will have the AI robots that will be able to, be like little crab robots that travel all over our spacecraft and fix them when they need fixing. But that day is not yet.
Is this how the Matrix started?
It's very possible.
Little bugs.
Yeah, what? Yeah. Other spacey news, though, that's not as positive, but could come out being positive. So SpaceX, back in 2015, it was part of the mission launching the Deep Space Observatory for NASA out to L2, and its second stage booster rocket got up into orbit around the Earth and did not have enough fuel to get itself back down to crash, to come back to Earth. So it was stuck in Earth's orbit. It didn't have enough fuel to have a controlled orbit, so it's been in a chaotic orbit. And now an old Falcon 9 SpaceX rocket is going to impact on the moon.
Okay.
It's going to run into the moon, and it's probably going to happen on March 4th. They don't know exactly because there are factors like solar radiation and other things that are going to impact the trajectory of the rocket. But at this, they think March 4th, it will land somewhere on the equator on the far side of the moon. And hopefully we'll have a bunch of our satellites that are around the moon in proper position to be able to look at the ejecta. And so that is the hope right now. But it's still being determined.
Yeah.
Chaotic orbit. This is what happens. These things are starting to happen. This is the first time that we have an unplanned or accidental impact like this on the moon. We have impacted the moon before on purpose. We've had things land on the moon also, but this is one of those things.
We just keep this is just more mass from Earth that we're never going to get back. We're just sending it away.
Yeah, I mean, we don't need that booster rocket. I mean, I bet SpaceX would like to get it back. I wonder if, at some point they'll send a mission to get It's their rocket booster parts.
Just so much mass. The telescope, all our satellites. We just so much mass in space that is no longer on our planet. It's crazy. I'm sure it's like 0.000000001% or something. But it's still, it's at a certain point, it's got to make an impact, right?
At a certain point, not yet. Unless we start mining asteroids.
Oh, and then we can bring mass back. I see.
Yes.
I see. Interesting.
Yes.
Huh.
We'll bring it all back. The universe, it will provide.
I guess. Unless we make ourselves too heavy and then that messes things. Anyway.
We don't need to talk about that. Why don't we talk about not future stuff, but some past stuff? What happened with the dinosaurs?
Do you know I can tell you what month the dinosaurs died?
No. What? So we're talking about like what month and where this rocket booster is going to hit the moon. But now you can, like, what is this? Astrology for dinosaurs? What's happening here?
Not exactly. I mean, we know it was about 66 million years ago at the KT extinction event, right? But what month? That's what we want to know. A recent study in Nature builds on earlier evidence to suggest that dinosaurs probably met their demise late May, early June. Yeah. Okay. This is looking at a site called TWIS, which is in the Hell Creek Formation in North Dakota. It's a place where there's just so much fossils to go through. And they've actually been able to observe the Cretaceous-Paleogene boundary in the succession of sediments. So they actually have specific elements in the sediment that leads them to believe that this is a flash point of exactly when it happened. And so they have fossils of early dinosaurs, early mammals, fish, plants, other things. They're really well preserved. They even have, some of them have soft tissues, skin, bones, and all sorts of things that they can use to kind of date these things appropriately. So the main reason they think that this caught the main moment when the meteor hit is the sediments. Okay, I'll just say the sediments. You can trust me on that. There's a whole thing, but the sediments tell them this is this. But also there were these things called melt spherules in the soil. So tiny glass balls cooled in flight from molten rock. So the idea is that when the asteroid struck Earth in the region of what we now call the Yucatan Peninsula in Mexico, it spread debris and melt spherules for thousands of kilometers. And so these being there tells us it's that exact moment. The third piece of evidence is Secchi waves. So they're standing waves in deep channels that tell them that this has to do with this larger impact. So all that to say, that means this is the extinction event in there. Now, how do you know, okay, this is the extinction event. Okay, this means it's late May, early June. They analyzed fish bones, specifically sturgeon fossils. And it turns out that sturgeons, they move from summer to winter. They have seasonal changes in where they live, if it's salt or fresh water. And so they have growth rings in their bones that show that alternation between salt and fresh water. They're in fresh waters in summer months, salt water in winter months. And so in the specimens they analyzed, this lined up with modern day sturgeon bones as well. The last growth increment matches the transition from spring to summer. This makes them think it's June. This does also confirm earlier evidence from fossil plants, water lilies, which I guess so at the point of impact, the lake froze, which preserved these fossil plants perfectly. And by comparing them to modern water lilies, they were able to see where they were in annual patterns, and it was late May, early June. So these two clues tell us the dinosaurs went extinct beginning of the summer.
But maybe like the beginning of the summer, when the summer was 66 million years ago, because climate change potentially has adjusted when that's happening.
And that is a great point. Yes. So you're totally right. We don't know if sturgeon movements were slightly different because temperatures were slightly different in the waters. we don't know if the same is true for the water lily. So you're totally right about that.
Yeah.
This is based on current modern day seasonal fluctuations.
It
So
occurred.
I don't know.
It occurred during what would have been
Yeah.
the same time period, with the same seasonal time period that the plants would have been doing their thing, flowering or not flowering or whatever. Yeah, that's interesting.
So I would say you're totally right. It's maybe silly to say that this happened in late May, early June. It is more accurate to say it happened in late spring, early summer. And you can use it to relatively date other fossils that have seasonal ties. So instead of saying this happened in June, this happened in August, you can say this happened the season after this happened.
Yeah.
So I don't know. This is... It's interesting, though. I think it's very interesting that you can, understanding how relative dating and radiocarbon dating and all these other fossil dating systems work. It's very cool to see this other method where you can pinpoint down to the season.
Yeah. So it's going the radiocarbon dating and the sediment layers, taking it down to, era or epoch. Like how many thousands of years ago was it generally? Plus or minus 10,000 years or 100,000 years. And then because of what we know about biology, you can start putting the things together. Like, well, plants seem to do this and these modern plants do this. And so, yeah, let's put all these pieces together. That's some good archaeological sleuthing.
Mm-hmm.
Yeah.
I love the idea of taking fossils and comparing them to modern day animals, and you can see similar physiologies and behaviors. Yeah. And speaking of that, I want to talk about ichthyosaurs.
Ichthyosaurs.
Giant fish dinosaur things from the late Jurassic, about 150 million years ago. So at this time, there were ichthyosaurs, which hence the point of the story. But also something really important happened on the planet. Two entirely isolated oceans, the Eastern Pacific and the Western Tethys, which would later become the Atlantic Ocean, came together. They merged across the Hispanic corridor. This merging pushed a bunch of aquatic animals into common space, into common food webs that had evolved separately in these separate oceans. And so you see some really bonkers evolution happening as a result of this kind of convergent, the union of these oceans.
Yeah.
And so a research team consisting of scientists from Colombia, Canada, and Germany explored the neotropics. They used the fossil record. They looked for all sorts of cool stuff and discovered a new species of ichthyosaur. It is named Kyhytysuka sashicarum. So Kyhytysuka. That's why my face did what it just did. So this is the first Cretaceous hypercarnivore ichthyosaur. Let's break down those words. So I already told you what an ichthyosaur is. It's a giant fish-like marine reptile. A hypercarnivore is an animal that only eats big animals. And Cretaceous, that means this specific time period. So the Jurassic ichthyosaurs, they ate some big stuff, but over kind of evolutionary time, their teeth shrank. They changed their purpose in the ocean. They kind of they ended up eating a lot of smaller fish and stuff like that, invertebrates, for about 70 million years. And then Kyhytysuka shows up, has re-evolved the capacity for hypercarnivory during a time when there was huge ecological upheaval, when these two oceans were smashed back together. So they have these sawtooth cutting teeth, they have piercing teeth, they have crushing teeth, they have the litany of teeth that you would need to eat large other animals. So a couple things on this, that's pretty much the whole story, but it tells an interesting story in that it is a situation where, we like to think about evolution as always moving forward and progressing towards a goal. That's not how evolution works. And I like this story because it reminds us of that. It reminds us that it goes back and forth and in and out. And, oh, that didn't work. Let's try this. And so this is a really good example of they had the ability to eat these giant animals. They specialized into these groups to eat smaller animals. And then they were put back all into the same tank again. And now it was time to start eating big stuff again.
It's like, oh, I'm going to grow some teeth now because food source.
Right. And of course, when we say that, I will just spell out in case, people are not super familiar with how evolution works. What happened was there were some of these guys that had slightly bigger teeth and slightly better teeth for hunting. They survived better. They reproduced more. And so over evolutionary time, which is long, it's, I'm talking about like it happened right then, but it didn't. It was a long process. But over time, these ichthyosaurs that were larger, that had larger teeth, that were able to eat larger animals, succeeded more. And so that became the dominant phenotype, the dominant type that you saw in these animals. So I love the push-pull of evolution, how fun that is. And also the idea of the push-pull of the tectonic plates and the fact that they were separated into their own little spots. And then the plates moved, they were pushed back together, and they went, oh God, back to that thing.
Okay, got to adapt. Let's do it. Yeah. I think that's one of the, I think that's the main point, is remembering that evolution is a process and it doesn't have a direction. It's just the goal is survival. The biological goal is survival. So how do you make
goal
that
one,
happen?
Don't die. Goal two, reproduce. That's it.
Yes.
That's a successful evolutionary trait.
Mm-hmm. And we find all the time that there are bits of DNA that have been silenced, but that can be unsilenced, that, they've just been in hiding. Chickens can have teeth. Yes. Who knew? Like this is thank goodness they don't, but they could. Those genes are still there. And it isn't just a one-off that's the reality. It's all of us. We all have potential. Mm-hmm.
Give me a tail. That's all I have to say.
You've got a little tiny one. You could probably grow a longer one.
I want a big one, but I also would have to turn on the genes to make it fluffy, because otherwise,
yeah, that would take a while.
Yeah.
I recommend maybe, it's like a tail that Velcro's on.
Okay. I could do that.
Okay.
Moving from tails. Tell me about frog legs.
Yes, moving from tails, let's talk about regenerating frog legs. This is one of the coolest stories this week. Like, that it goes from the idea, so science fiction future, you can grow whatever limbs you want, right? This is like a dream of science fiction. Yes, Blair, you can have a tail. But in the more near term, it would be amazing to be able to regrow limbs for people who have lost them for whatever reason. So amputation, for, various reasons, disease, trauma, whatever. Right now, we don't have that. We have prosthetics, and the prosthetics are getting better and better, and their neural integration is getting better and better. But that's one different path. The other path is, if you've watched The Expanse, one of the even many science fiction books and movies, there's the idea of just growing a new arm, that we have the technology to get the body to do what it knows how to do and just grow a new arm. We knew how to do it once. Why can't we do it again? And so in this study published in Science Advances, these researchers have taken multiple drugs related to growth factors that have been shown in multiple studies to work in a variety of ways, and used the idea of organisms like the salamander and others that can regrow limbs, looked at how that happens in other organisms. But in this, they've looked at frogs, Xenopus laevis, and these frogs, they don't regrow limbs when they're in their adult form. As tadpoles, they can regrow all sorts of stuff, but as adults, they really don't regrow anything. And they've created what they call what they call the biodome. And the biodome is like it's just a little plastic cap that they fill with all these growth hormones and factors. And they stuck it on the end of these frogs' amputated limbs and looked to see what happened in the regrowth of the limbs. And they found that 24 hours of application of the biodome to the frog limb resulted in the propagation of what ended up being 18 months of growth. So over 18 months, these limbs grew to the point, with all these growth factors, they grew to the point that they started regrowing bone and the little digits at the end of the hind foot. 18 months of growth, and it wasn't a full regrowth yet, but it began the process. It restarted a process, just 24 hours of application of these neurotropic growth factors and growth hormones and different things. It set the stage for the biochemistry for the cells that were already in the damaged part of the leg to, instead of forming a scar tissue, start forming new tissue. And the growth continued, like I said, to the point that they started growing digits at the end of the leg. They also showed that compared to a normal limb that had not been amputated, that at the end of 18 months, the limb was strong and actually had a significant amount of, could withstand a significant amount of force. So the bone, the tissues, everything were growing and healing. This isn't, it's not like 100% done, but it is a step along that path towards.
So I just want to confirm,
yeah,
what I'm hearing. So 18 months left alone, it would grow?
Yes. So 24. So they have this little cap.
Mm-hmm.
It's like I don't know. It's like if you when you go and you put a quarter in one of those, like you go to a pizza parlor with a toddler and they want to go get one of those little erasers out of the machine,
yes.
And you get a quarter and the eraser comes in that little plastic cap thing.
Uh-huh.
It's like it's like that kind of a container, except they've set it up and filled it with all sorts of biological growth things, and they put it on for 24 hours.
And then it takes an additional 18 months for it to grow.
Yes, but they didn't reapply it. Just 24 hours.
So it's giving it a jump start.
It just jump started it. Yeah.
Okay. And this is because frogs normally cannot grow their limbs, but other amphibians can.
Yes.
So this is where it takes a pretty big jump if we're trying to do it in mammals, is that amphibians have, you're talking about kind of like these latent genes. Yeah. They have them very closely related.
Right. So this is a big question. They chose the frog because it was closely related to regenerative amphibians, but not regenerating. That lack of a process in itself is closer to mammalian biology. So that's why they chose the frog in this particular instance. But now next steps are to see whether or not they can do similar work in mammalian models.
Yeah, throw the biodome on a mouse. Go for it.
Can they regrow bone? Can they regrow all this stuff? Yeah,
so in this sci-fi kind of next step, you would go to the doctor, you would get your 24-hour treatment, and then you'd have your limb in a like a sling or something, nice and covered, and then over the next 18 months it would get bigger and bigger.
Right.
Wow.
Probably, you'd probably have to wait a couple of years for it to grow back, but it would potentially grow back over time. I mean, the question is the timeline something that humans in our lifespan, could we live with that, right? Could we accept that timeline? People want everything now, right?
And because we're longer lived, is it not the same? Would it take 5 years or 10 years in us instead of 18 months? Because of that, and also because our biology is pretty complex. And
I mean, I do love the idea. I love the idea of somebody who's like, super buff on one arm and the other arm is a baby arm. It's like the 2-year-old toddler equivalent because it's been growing for 2 years. That is the question.
That's what I was thinking about too, is how it would grow. It'd be so interesting to see how it would develop. Wow. Anyway.
Yeah, so many questions. I mean, the leap from a frog leg to any application in humans is a massive leap, but it's an interesting proof of concept, and we'll see where it goes. Yeah.
So, food for thought. Would you cut off a limb in order to have a new one in 2 years?
I would not, because my limbs are fine.
I know. I can see also that being a thing, right? Like, instead of getting this elbow surgery, I'll just cut it off and I'll get a new one in 2 years.
Well, that's the next step of could they advance the technology so that it makes it worth it for somebody?
Right.
Yeah, there's a lot that would go into that.
Wild.
We'll see. There's a lot still to be seen there. And then moving on from frogs' legs, I want to talk about some really deep life.
Yes, please.
Yeah. Oh my gosh. So this to me it boggles my mind to imagine. Can you think of life in the deep ocean? Mm-hmm. When you think of life in the deep ocean, like, what do you think of?
Jumbo octopus.
Octopuses, yes. But you think of like the deep hot smokers, the vents where there's a lot of bacterial life, where there's a lot of nutrients coming out, and there are these hot spots of lots of biodiversity and a lot going on. But then we talk about the bottom of the deep ocean as a desert. Mm-hmm. It's just like it's the spaces where there's nothing going on.
And that's why, like, when a whale dies and it goes to the bottom, it's like, because suddenly there's food.
Exactly. So a group of researchers in 2016, upon a Japanese scientific drill ship called the Chikyu, drilled 1,200 meters below the seabed. So they went looking at the bottom of the ocean, and then they drilled past it, 1,200 meters below, to see if they could find life, to see if they could find anything, any biodiversity in the crust of the earth below the ocean. They found stuff. They found like not very much life. Like they were finding maybe like just a tiny amount of life. Not very many cells, where there would normally be hundreds of thousands of cells in any topsoil, millions of bacteria, microbes in a spoonful of topsoil. Here they're finding, 500 microbes, like the smallest factors of factors of difference. And so they tried to grow them, and they tried to find out about these the bacteria that are down there. They're like, how can bacteria live down there? What's going on? And what they determined is 1,200 meters below the surface of the sea, below the seabed, it's getting hotter and hotter and hotter because you're drilling through the crust toward the mantle. And so even though the bottom of the ocean is cold, below that seabed surface, it gets hotter and hotter and hotter. And so life that exists 1,200 meters below the seabed is living at about 120 degrees Celsius. So it's extremophiles trying to survive. How are they surviving? We don't really know what they're living off of acetate and hydrocarbons that are in the crust of the earth. they're living off these like just toxic things normally, but they're surviving in the crust. And in order to survive, because they're being bombarded by radiation from the center of the planet, by the high temperatures, by so much stuff going on in the soil, they found that the metabolic rates for these bacteria were super high for the biosphere. Basically, these microbes who live in the deep, deep crustal seabed, their hypothesis is that these microorganisms maintain a really high metabolic turnover to undo all the damage that their environment causes. So they're not in a torpor like like a tardigrade could potentially, be in a torpor state to, withstand it. They're alive and they are living really fast because they're like, oh, I gotta fix all that DNA damage.
Oh my God.
So it's a very, I think for the microbial deep seabed crustal life, it's a very lonely and just intense kind of microbial life. If they're conscious of what's going on, they're very, very high strung.
That's crazy.
Yeah. Yeah. So these researchers, they've just published their work this last week, and it's in Nature Communications. Rapid metabolism fosters microbial survival in the deep, hot subsea floor biosphere. Deep, deep life. It's down there.
Keeps on going. Next, we're going to find it in the magma.
Just amazed that something could live. Right. Why not? Why not?
Yeah.
Just why not? Okay, there's life down. Okay.
Magnaphiles.
You shouldn't be able to live down there, but you are. Okay.
Yeah. Yeah.
We'll see.
I did the conversion if anyone was interested, because I used to be able to do the conversion pretty easily in my head, but I can't anymore. It's about 250 degrees Fahrenheit.
It's hot.
It's hot.
You would not be happy. 120 degrees Celsius is very hot.
You'd be dead.
People would be dead.
Yeah.
We're not liking it very much. So these little microbes living at the absolute edge of what should be possible for existence, their metabolisms are doing everything they can to make that possible. If you guys could just back off a little bit, it'd be cool too. Okay. This is This Week in Science. Thank you for joining us for another discussion of science and learning and curiosity. If you're enjoying the show, please tell a friend. Want to come back for some COVID conversation?
Might as well.
Let's do this. Oh, you know what? There's a new variant.
Oh.
Oh, yeah. Well, it's not like a variant. It's an Omicron lineage variant.
Okay.
And so this is where the fun starts. It's like a subvariant.
So does that mean everyone who got sick with Omicron is okay? Because most of the world
likely
got Omicron at this point, right? So
yes. So that is the good news here. The Omicron lineage has been taking over, and the main lineage for a very, very long time was called BA1. And suddenly, there's a new one that was detected called BA2. And BA2 is even more transmissible than BA1, which is like, okay. And so it is rapidly outpacing the original Omicron, but it doesn't appear to be causing any worse symptoms. So
they're just trying to get all the ones they missed. They're just playing cleanup at this point.
Yeah. So at this point, it's a sublineage. It's related enough, very closely to the original, the OG Omicron, so that antibodies, T-cell response, all that stuff is still going to work. So it should not be, it's like, it's still Omicron, more or less. Just a slight variant. Little bit easier to catch. I don't know. But it's here in the United States. No,
of
United
course
States. It's here. And the World Health Organization president is saying we really need to be ready for more variants. There will be more variants because we know that mutation in this virus happens very readily. So as long as it is spreading between any individuals, there's the possibility of more mutations and more variants.
This is the price of us not shutting down and doing things.
Yeah.
So it allows for things to spread and mutate.
Yes. But good news, Pfizer has started a human clinical trial of its new Omicron-based vaccine for COVID-19. So it has switched up its chemistry, really. So instead of the old alpha spike protein, which everything was targeting, now this new vaccine, it's a vaccine variant, really. Is made for the Omicron specific mutations so that it'll be much better. They've started testing it on around 1,400 individuals. They're going to be testing it on people who have not had any vaccines and also people who have been previously vaccinated to see how it mixes with previous vaccination as like the next booster shot that would be given possibly beginning this spring, summer into the fall.
So question about that.
Yeah.
Would the idea be to give everyone that booster? Or would they like test you for Omicron antigens and then go, no, not you. Or would they say, did you get COVID from this date to this date? Okay, not you.
And I think this is one of the big debates that we are, that our health systems are not dealing with well, which is if you've been infected, the immunity that you have gotten from infection, how is that going to mix with future vaccination? We also don't know a lot about, how long it takes for immunity to wane. Mm-hmm. For this newer Omicron variant. Is it different from Delta? Is everything waning in the same amount of time? As it stands currently, people are suggesting that if you've been infected, that similar to like your last vaccine dose, that you wait at least 6 months to get a vaccine if you're going to.
Right. But then the problem is with Omicron that so many people got it and didn't know that they got it. Because there were some people who were asymptomatic, went about their lives, never got tested at the right moment, spent their 2 weeks with Omicron, had no idea.
No clue.
Or
right.
5 days, I guess, whatever. Anyway. But I think that's what makes this really tough is that up until this point, asymptomatic cases didn't happen a lot. They happened, but they still, the people who had strong immune responses were usually the people who got really sick. So this makes it such a weird new world in that everybody was getting it and everyone was having varying levels of responses, whether they got sick or not. The amount of antibodies you would have developed would be varying depending on when you got vaccinated and boosted. It's kind of a logistical mess.
Yeah. I think looking forward, statements made by the head of Pfizer are not on this kind of like, oh, we're going to be like on a every 3 month or 6 month booster schedule. It's not like we want to have a booster for each and every variant that comes out. Like we want to have it like constant. We don't want that. And Pfizer said that's not what they're planning on. They're thinking, looking at mutation rates and looking into the future, that it, like we said before, might be similar to the yearly flu vaccine. And maybe they will be given at the same time so that, whether or not you've had it, you go in for your yearly vaccine and then, you're taken care of and you just go ahead with your life because you've done what you can.
Yeah. I could see for this one, though, them saying if you didn't test positive at any point, you get first dibs.
Jump in there. Yeah. Well, they're definitely, again, going to focus on people who are immunocompromised. They're going to focus on the elderly. They're going to focus on the people who are at highest risk for infection and for the severe outcomes of it. Mm-hmm. But it's good to know the clinical trials have started. The process is going.
That's great. I'm glad the system is moving quickly.
The system is moving. Yeah, which is, it's working. It's good. It's good to know it works sometimes. Yeah. But tell me about the COVID mice.
Oh, sure. So remember how we were like, oh, don't worry, COVID can't spread to any animals. Oh, no, wait, actually, maybe sometimes cats. Oh, no, wait, turns out maybe weasels. Oh, wait. So, we kind of had to backpedal on that anyway. But a team of biology researchers from Georgia State University recently found that some of the newer variants of the virus that causes COVID-19 can infect the respiratory tract of wild mice. The original strain could not. This was looking specifically at the alpha and the beta variants. And so they were the first two. Yeah, they were the original ones. And they were able to replicate in the lungs of wild mice. And so what this means is, on the pro side, that you can use lab mice now as a model for researching the virus, which is really good news, actually. Of course, on the bad side of the coin, there is a higher potential for the virus to replicate in rodents, which means wild mice and rats in cities and stuff, living in close proximity to humans. So that does raise concerns about bats, rodents, other wild animals, that there may be a situation where they could not only replicate and hang out in mice, but also they could mutate within mice and then jump back to humans. Public health experts and researchers generally currently agree that infected animals have likely gotten the virus from people and not, from each other. There's a low risk of transmission from animals to animals and even lower from animals back to humans. So they really don't think that that's any cause for panic yet, but that is something to consider moving forward. Next, these researchers are going to look at other variants like Delta and Omicron in mice to see if they can replicate as well.
Well, I'm going to say it probably will be able to.
Mm-hmm. I would agree.
That's my hypothesis, and I'm going to just stick with that. Yeah, I mean, we've already seen hamsters. Mm-hmm. And there's I think Hong Kong, they had one outbreak that was tied to a pet store employee at a pet store where all the hamsters had COVID. Mm-hmm. And so it's a question whether it came from the hamsters or whether the employee had gotten it and then given it to the hamsters and other people. Big question, but ferrets and hamsters and cats and dogs, all these pet animals that we like to spend time with. Yeah. And animal reservoirs are such a huge issue. I mean, I love the possibility now of mice, our model lab animal. Let's do research. That's great.
Yeah, we still have so few medicines for COVID that could be a huge benefit, actually.
Yeah, absolutely. But the downside, like you said, is like, okay, so the mice and the rats in the city where you live, if they all have COVID and we get rid of it in the human population, but it's still in that reservoir, what happens when it pops out again? What does it look like when it pops out again? Mm-hmm. And that's the big concern. Researchers are really busy talking about the concerns of animal reservoirs, human reservoirs, even like patients with suppressed immune systems, like cancer patients and others who have become basically reservoirs for mutations to occur. And whether or not it'll allow for enough mutations to lead to the point that a variant will evade antibodies, evade vaccines, and evade the T cell response. Because where we've been right now is you get vaccinated, okay, you can have a breakthrough, but your T cells are still primed. And your T cells are your immune system's going to protect you and keep you from having severe disease, most cases. But if it gets to a point where the mutations allow it to evade the T cells, that is the huge concern. But we don't know. Nobody knows what's coming next. We keep looking, and they're looking at the various mutations. There are researchers who are very worried that like we talked about in the last section, evolution doesn't necessarily need the virus to become less virulent. It could become more virulent. It could switch up, and that would not be great for us. But we can just keep following the genetics, which a lot of these, we have monitoring groups all around the world that are following the new variants, looking at strains as they come up, see what's going on, and tracking them so that they can try to make predictions about where things are going to go. But as we've seen already, there have been variants that were variants of concern that researchers thought were going to become a really big deal that didn't at all. They didn't go anywhere. And so right now, where we are, Omicron, BA1, BA2, the wave is making its way quickly through populations. Hospitals are getting overwhelmed, but it hopefully will not last too long. And fingers crossed that nothing really big pops up anytime soon. I mean, it would be great to have let's finally have our hot vax spring and summer. But vaccine immunity and Omicron spread are very promising for the pandemic becoming endemic at the moment.
Which, how crazy is that? That's our hope at this point. And that 2 years ago, we're like, we just don't want to make it endemic. We just want to get rid of it. Now it's like, can you just chill and hang out in the background? That would be great.
Yeah. Can we just have 2 years and not 5? Can we just, come on.
Just go play with the flu in the corner. We'll call you when it's time to come back. Okay.
Yep.
Just go take care of yourself. Just play amongst yourselves.
Go home already. Go home, coronavirus. Go home. Well, I think there's good news, though. I think there's room to be optimistic, even though there's a lot of things still pandemic-wise that we're all tired of. But I do believe that there's, I don't know, depending on your personality. I'm not going to say get your hopes up. I don't want to dash your expectations later. But
yeah, I also do just want to mention a lot of people died, and that sucks. And a lot of preventable death, and that's the worst. And it feels terrible. So if we can just stop more preventable deaths from happening, we'll take the loss at that point. It's still terrible. Families have been ripped apart, but can we just stop? Enough. Fingers and toes crossed. Things
right.
I
know. Every week it's like, where are we now? Have a little check-in. Where are we now? But you know where you are right now? Where? This Week in Science. Yes, that's right. This Week in Science. We come to you every week to talk about the science news from the past week. A lot of science, some discussion, a lot of curiosity. We hope that you enjoy the show. If you do enjoy it, head over to TWIS.org and click on that Patreon link. Patreon is how we support the show. You can become a supporter. Choose your amount of support, $1, $10 a month, anything $10 and more a month. We will thank you by name at the end of the show. We really can't do any of this without you. Thank you so much for your support. And now, it is time to come back to the wonderful world of happiness and animals with Blair's Animal Corner.
She loves our creatures, great and small. Biped, millipede, no pet at all. If you want to hear about animals, she's your girl. Except for giant pandas and squirrels. There's Animal Corner.
What you got, Blair?
Oh, well, I had to bring a story about hippos. So
yes, you did.
Watch the clock. Don't let me talk too long. But I will say, Kiki, you should pull up the story because there's some sound in this that I want to play.
Will do.
So I want to talk about hippo calls. Did you know that hippos call? They have fun sounds that they make?
I figured they were like, They can,
Kind of.
open their big maw.
Yeah, pretty much. Well, they have what's called a wheeze honk call, and it's very, very, very loud. It can be heard over long distances. As we've discussed many times, hippos are territorial, but there has not been a lot of research on their calls in particular and how that kind of folds into their territoriality, if that's a word. And so a new study that came out this week looked at hippos and how they recognize each other's voices, how they respond to hearing other hippos, and if it matters if it's a stranger, a neighbor, or someone from their own group. This is the University of Saint Etienne in France. In France.
In France.
In France.
Yeah.
Francais. And the study highlights that hippo groups are territorial entities that behave less aggressively towards neighbors than strangers. So you might think actually that there would be more friction in between neighbor groups because they have to share resources and they're maybe, males are sneaking in from the other one and it's a whole mess. But actually, they're used to tolerating each other. And so it looks like they are less aggressive towards the sounds of their neighbors. This study highlighted that hippo groups, they have kind of a different response to a stranger versus a neighbor versus someone from their individual group. And so they recorded calls from each hippo group. They played the recordings back to all the other hippos to see how they'd react. So this is not a lab. This is not a zoo. This is in the wild in Africa, in Mozambique. They went to a reserve and they played these calls to see what the hippos did. Yes.
It sounds more like a bird than I expected.
I love it so much. But so they would play these recordings back and they wanted to see if there's a difference in the way that they performed to familiar versus outsider sounds. They found that hippos respond to hearing a played back call by responding vocally, approaching the sound, or spraying poop. As they are wont to do, which is a territorial display. Interestingly,
I love them.
The response varied depending on whether they were hearing hippos that they knew or ones that they didn't. The overall intensity of the response grew when they heard a stranger, so they responded kind of louder, bigger, more aggressively, larger movements if it was a total stranger they were hearing. They were more likely to spray dung, which again is a territorial behavior. And so the thing that's really interesting about this is that, like I said, hippos are really territorial. We've studied all sorts of things about their territorial nature, but this has not been thoroughly studied, the way that they respond to sounds. We know they make sounds, but there is this kind of understanding that because hippos just kind of lay there in the water during the day, they're very inactive, they sleep, They have their nose and their eyes and their ears poking out, but otherwise they're pretty much submerged. It looks like they're not up to much. They could be totally conked out. But these results show that they really are paying attention at all times of day to their surroundings. When they heard a hippo played from the shore, they responded right away. It didn't matter if it looked like they were dead asleep. They heard another hippo, they responded. And so this has implications for hippo management and other animal management. This is maybe time to look at how a lot of other animals that hang out in groups that have neighbors and stuff, how they interact, because they'll relocate a population to make sure that they're healthy sizes, that there's proper resources, all this kind of stuff. But it might be a good idea to play sounds of the group coming in to the neighboring groups before they arrive. And it also might be a good idea to play sounds from their new neighbors to the group they're about to move.
Right.
So that they're more familiar. So, because you would be able to see their response kind of calm over time when the sound became more and more familiar. And that would be your cue that it would be okay to move them at that time.
They know what they sound like. So, oh, you're not a stranger anymore. I've heard you. I've seen you across the way. Auditorily.
Exactly.
I feel like I know you, in fact.
Yeah, exactly. It's like when you meet a celebrity that you're like kind of obsessed with and you act like you've known each other for it's the podcast
That
thing,
happens
right?
all the time.
No. This is like the podcast thing, right? Especially podcasters. I guess celebrity is a strong term. But if you listen to a podcast every week for, I don't know, like 90 minutes, you might start to feel like you know
For 21
these people.
years.
Yeah, exactly. And so when you meet them, if one of our listeners meets us, they might feel like, I know them already. They're my friends. And it's because they've been listening to us this whole time.
Hippos are the same.
Yeah.
So basically, it's like podcasts for hippos.
There you go.
Yeah.
And so in future studies, researchers hope to learn more about what hippos are communicating with their calls, if there's specifics to what they're saying. And they'd also like to explore how they recognize the sound of other hippos. What about their voices give away that they know them or they don't? And do those voices give away other characteristics like size, sex, or age? We don't know any of this yet. So there's a ripe vein of research to be had related to hippo calls.
Oh, Blair, you're so excited about the future.
I'm really excited.
Of hippo vocalization science.
That's a guarantee that when that study crosses my desk, you'll hear about it.
Awesome.
Yeah. And jumping just across the ocean there to South America, I want to talk about jaguars.
Okay.
So on Valentine's Day 2020, in Colombia, some jaguars were observed mating, doing their whole like mating dance, doing growling, fighting. And it's a 2-day process for these jaguars to kind of, it's a will they, won't they scenario for 2 days with sometimes will they's sprinkled in there over the 2 days. And so they observed this. What was strange was that right before that, that same female jaguar was spotted playing with her 5-month-old cub. But during this Valentine's Day romp, the cub was nowhere to be found. This is a problem because in jaguars, not to mention lions, pumas, all sorts of animals, but definitely a lot of wild cats, infanticide is a thing. So infanticide means that a male will kill a baby so that the female then can go back into estrus and he can mate with that female and then she can have his baby instead. So it's very sad, but evolutionarily, it is a successful tactic. Otherwise, this male would have had to find another mate or wait a whole nother, however long, usually a year, right, to be able to have a baby. And if you're trying to win the game of evolution, as we were talking about earlier, you want to get the baby out now. So this is the benefit from the side of the male to infanticide. Again, very sad, but nature can be sad. So knowing that, people watching these jaguars were upset. Oh man, this means the 5-month-old was probably killed by the new male. A few days later, mother and cub reappeared. So that cub disappeared just long enough for mom to have this 2-day romp with this boy. So what was happening here?
Where was the young one? What happened?
Well, other big cats, like lions and pumas, have been known to hide their young during other mating to prevent infanticide. So hiding is a beneficial tactic to prevent this. But this is an extra level because it looks like the female engaged in this kind of flirty 2-day love fest to either make the male think he was successful in impregnating her, which he couldn't have been because she was nursing the cub. So that's not biologically, that's not going to happen.
Tricky.
So this kind of like getting him off the scent. Like, oh no. I'll have your baby. Just kidding. So there's this idea. There's the other idea that like you could hide the baby, do this, And then if the timing works out just right, the male could think it was his.
Right.
So this is something that has never been written down in jaguars.
But this happens in other cats, big cats.
Yes, it does, but not to the level where it looks like the female is engaging in this intense multi-day flirtation. Because in the jaguars, they have mock fights. The female bares her teeth. She makes vocalizations. And so this is kind of this intense mating ritual with lions, for example. It's a lot less fancy because there's just a male and a bunch of females. And if he's the head male, then that's it. You just,
right.
He's going to do what he's going to do. So I think this is unusual because of the kind of the amount of effort that the female had to put into pretending that she was an estrus. So this makes it pretty unusual. Again, researchers had no recorded history of this, but so they asked around and they found 2 other eyewitness accounts of this happening. So now they have a sample size of 3-ish.
Huge sample size.
Yeah, so this is a very small sample size. However, this is something that hasn't really been looked for. And the other problem is that jaguars are notoriously great at hiding their babies. So how do you know the difference between just seeing 2 jaguars having a fun couple days that could result in a baby and a female who's hidden a baby who is pretending to engage?
And you have to be watching the animal the whole time. Yeah, you have to be tracking them. Yeah.
Yeah. So that's kind of the difficult piece here. Now, as there is less wild space and humans live closer and closer to wild animals like this, there is a chance for better accounts as time goes on, especially if you know what you're looking for. Locals could see this happening because they see the same animals all the time. So a lot of the time, if you live around wild animals, you will get to recognize individuals. And they would be able to say, I saw that female 2 days ago with a cub, and now she's with a male. So there's that possibility. There's also the possibility of setting out specific research trips looking for this during specific seasons, and they can catalog and they can use cameras and they can do all sorts of stuff to kind of track individuals and maybe figure this out. But the other problem is that as, again, wildland disappears, that means that there is potentially less space to hide babies. So that is also something that it's important for us to understand as conservationists, because if conservationists can go out there and make sure there are spaces for babies to hide, assess a space, make sure where jaguars are living, those spaces exist, then that can reduce infanticide. And obviously you don't want that for a bunch of reasons, but the main one being if you want to boost a population, you don't want to, first of all, let one bully male father too many babies, but you also just don't want to risk another birth and early life in a baby if it's doing fairly well, because there's bad odds there. If you had a successful baby come out, mom didn't get hurt, baby didn't get hurt, and baby grew to be some weeks old, you want to continue that trend because that means that some pretty important hurdles have been jumped. So it's tough out there.
It is tough out there, and how can we make it easier? What can we do as humans? We're not doing what we can necessarily to help the animals in the wild world, are we?
Yeah. Step 1 would be to not cut down rainforests and destroy jungles and keep wildlife refuges protected, and to not start forest fires and all that good stuff. But otherwise, also don't traffic wild animals and all that stuff. But also, if you're trying to, if you're trying to reduce the amount of disappearance that's happening, but if you're also trying to add back, if you're trying to augment the population that exists and increase it, then you need to understand the reproductive history and patterns of a species. So this is really important information.
Yeah. The indigenous knowledge aspect of it, I think that you mentioned, is really important as well. We don't think sometimes about the number of animals that there are in populations and how they interact with human populations, especially in the jungles as humans are encroaching, and and also the tribes that have lived in the jungles forever.
Yes,
how they know it
successfully.
so
Yeah.
successfully. Yeah. So there's a lot
Yeah.
to be learned there and brought into this whole conservation game. We'll see.
Mhm. Yeah.
Love it. I love it. Sneaky mother jaguars.
Yeah. It's
Yes,
like, oh
go
yeah,
girls.
don't worry about it. Yeah, this
Right.
is yours.
Don't eat it.
Uh-huh.
Yes, don't eat it. It's
Don't
okay.
eat. Don't eat the baby. It's your baby, I swear.
Swear. Swear on, I don't know, the place I hid it. This is This Week in Science. I'm going to jump into my last couple of stories, which are so sweet. They're so sweet. Well, they're all about sensing sweetness. Indeed. Researchers have just reported in ACS Applied Materials and Interfaces their extra special, ultra sensitive bioelectronic tongue that measures sweetness. They had previously reported on a tongue that they had created to sense bitter flavors, and they decided to try again. Oh, no, it's not bitter. I apologize. That is exactly wrong. They had made an umami sensor. That's what it was. Umami. I got reports confused. But they'd made an umami sensor, and in that umami taste receptor, they had used the protein at the end of the umami taste receptor in the human tongue and applied it to this bioelectronic application. And they thought, hey, maybe we can use the same concept to be able to detect sweetness. And so they have made a sweet sensing bioelectronic tongue that uses what is called the Venus flytrap domain of the taste bud. So human taste buds, the sweet taste buds, have these two complex structures that bind to sugars. And the outermost portion of one of these structures is called the Venus flytrap domain because it's hinged and it opens and closes and it looks like a Venus flytrap. And it is activated when somebody consumes sugar or sweet things. And so they used that same protein, the biological molecule, and they grew it in bacteria. So they added it to bacteria, got the bacteria to grow this Venus flytrap domain, and then they added it to gold electrodes with carbon nanotubes, and they made a transistor device. So these bacteria have the Venus flytrap domain that can bind to sugars, and when it happens, it causes an electrochemical cascade that then goes from the gold ions to the carbon nanotubes and can actually transduce charge so that they can tell if something sweet has passed by this fake tongue.
Okay, so why?
Why make so robots can eat food? No, that's not why.
There has to be a why. There's a real why.
The real why is that it could sense the actual sweetness level of drinks. So it could be a sensor that could determine for, say, people who are diabetic or have other glucose intolerant type disorders, metabolic disorders, and have to limit the sweetness that they are exposed to. That there could be a sensor that you could pass, drop your food past, and it could let you know if it's an acceptable sugar quantity based on the sweetness. So something like a bioelectronic tongue could also be used in industry for quality control to ensure that everything that's coming out of a bottling plant is coming out at the right concentrations. And, to allow robots to taste someday.
Okay.
Because, and I just want to tag that on there because I know it creeps you out. So
Yeah. Oh boy.
Yeah, so I love this for the application. They're taking biology, they're taking what's already been created, what we've got in our bodies. They're like, hey, we can take this protein and we can put it in this bacteria, and the bacteria can grow it, and then we can use it in this way. And look what we did! Ta-da! So just the combination of engineering and biology and chemistry and electronics, like it all works together, and it's a really cool application. But moving on from bioelectronic sweet sensing tongues, how does our gut know when we've eaten something that's real sugar versus something that is artificial sweetener?
Why would it need to know?
Right? But why would it never maybe had to know evolutionarily, but there's been a lot of evidence that suggests that the metabolic response to artificial sweeteners is not exactly the same as to sugar. When we intake artificial sweeteners, there's a lot of evidence that our whole physiology, our brains, we react differently. We know that it's artificial sugar and not real sugar. How is that and why is that?
Mm-hmm.
And so some researchers wanted to look into this, and they're like, hey, let's get into the gut a little bit. And in their study, they've gotten into this part of the gut, and I'm going to put a video from Duke University on the screen here, but I'll try and explain it as much as possible for everybody in the podcast, of course. And so in this situation, there are nerves. The vagus nerve gets stimulated by activity in the gut, and the vagus nerve sends information about what you've eaten, whether or not you've had sugar, up to the brain. And then that gets your brain to do certain things, right? So these digested particles of stuff interact with the lining of your gut, and the lining of your gut has a whole bunch of specialized cells in it. A lot of little cell types. There are goblet cells, enterocyte cells, and one of them is an enteroendocrine cell, which they like to call the neuropod, because this one interacts with the nerves that go to the vagus nerve. So these enteroendocrine neuropods are really important for that information transfer from the gut into the brain. And what they found in this study is that when the animals in their situation, when the gut was given sucrose, plain sugar, it stimulated the neuropods, and the neuropods sent signals up to the brain. When they stimulated with sucralose, it did not stimulate the vagus nerve in the same way. It stimulated different receptors that stimulated an ATP response that stimulated completely different nerves that went on to stimulate the brain. There are two parallel pathways they've discovered within the gut for sugar or other sweet stuff. And our gut knows the difference and tells our brain.
What does our brain do with that information?
Well, it has to do with energy, right? So what have you eaten? How much energy do you have? How much energy do you have to spend? How much energy do you need to, how much blood do you need to send to the gut for digestion? What are the other things that need to go on? So the entire, your entire metabolism is impacted by what you eat. Now, what they don't know, and they haven't looked into it specifically, is how are these neuropod cells stimulated by fat, by salt, by other nutrients, and what do other nutrients do to this gut to vagus to brain pathway, and how do they affect that? But in anesthetized mice, it's this really interesting situation where there's a completely different receptor that gets stimulated with glucose than what gets stimulated with sucralose. And there's different pathways.
So I know there's a whole urban myth or whatever that artificial sweeteners make you more hungry or make you crave more sugar or make you gain weight. So I wonder if this has something to do with that.
That's what I was wondering too. Yeah. I mean, that's like one of the big questions is this why we have these different effects where your body goes, I got something that's like sugar, but it's not. And now I used this ATP. So can I have some sugar? I need energy. Can I have more energy? Yeah.
You just wrote me an IOU. You owe me.
Right. But it didn't stimulate the body. It didn't stimulate the brain in the right way that for what we've evolved to expect. So it's expectations being being let down, unmet expectations of the gut-brain axis. This is our hypothesis. Yeah. One of the really cool things about this study and the way that they did the study is they moved just past the simple like sucrose to sucralose and what's it stimulating. They used also optogenetics in the gut. So we've talked about optogenetics being used in the brain and how, you have to have these wires and how there's this huge setup. And so to think about, oh my gosh, how did you do that in the gut of an animal that's moving around where the gut is moving and everything, it's shifting around all the time. The brain doesn't move as much. And so everything's a lot more stable there. They created a whole new flexible wire technology to be able to use optogenetics to turn vagus nerve activity on and off in the mice while they were doing the experiment. And so one of the big things that, even bigger than figuring out how our gut and our brain work to, help us understand how our body even thinks of how our body does nutrition, right? How it does energy allocation and digestion. Beyond that is this new development in optogenetics could even push work further forward, because if we can do flexible wires in the guts of mice in a lab, who's to say that we can't do optogenetics in people, which has been a huge limitation. So
I just assumed they use those cows with holes in their sides.
Right.
So they got it done.
No.
But this seems better.
Even better.
Yeah.
I mean, that would be an easy way. What's going on in there? Open it, close it.
Yeah.
Nice cow fistulas. Cows are fantastic for their multiple guts, their multiple stomachs that allow.
Just walk around with a hole.
Yeah.
Totally fine.
It works for some, not for others.
Right.
But yes, the research moves forward. Do you have anything else to talk about tonight, Blair?
Not at all.
Not at all?
No.
Not a thing?
No.
Not a
I got to talk about hippos. I'm happy.
I think I'm done. Flirty jaguars. You're really good at that.
It's almost like I listen to it a lot.
I have a feeling. I can imagine you sitting there clicking the play button over and over again.
Yeah. You can ask my roommate. He heard me do it for quite a while yesterday. Your roommate and your roommates, your dog. Oh yeah. Sadie didn't like it very much.
But they'll get used to it. They'll be friends eventually. It'll be fine. And then you can introduce a hippo to the house. It'll be fine.
Yeah, it's fine. That's the only thing preventing me from doing it right now. So
how do you bring a hippo home? Well, first you got to play the calls at home a few times. Get everybody ready.
And then you give the hippo a cookie. But if you give a hippo a cookie, then he's going to ask for some milk.
You will. I knew where you're going there. If anybody has any questions about anything, you can send us your questions. We'd love to answer them, to talk about the things that you find curious as well. So let us know if you have any questions you want us to answer. Thank you so much for joining us. I hope you enjoyed the show tonight. And I don't know when Justin will be back. I'm sure we will hear how things are going as days move forward. Once again, congratulations to Justin. Everyone, the whole family for the baby.
He's either asleep or he's watching this right now.
No.
Those are the two options.
If he's watching right now, I hope we get a comment in the chat room. All right, everyone. Thank you so much. Shout outs to Fada. Thank you for your help with social media, with show notes. I know all the frustrations you're going through. Gord, Identity 4, R and Laura, everyone for manning chat room stuff. Identity 4, thank you for recording the show. Rachel, thank you for editing the show and your assistance. And I would really love to thank our Patreon sponsors. Thank you too. Richard Badge, Kent Northcote, Rick Loveman, Pierre Velazar, Ralphie Figueroa, John Ratnaswamy, Carl Kornfeld, Karen Tozzi, Woody MS, Andre Bassett, Chris Wozniak, Dave Bunn, Vigard Chefstad, Hal Snyder, Donathan Stiles, a.k.a. Don Stylo, John Lee, Ali Coffin, Maddie Perrin, Gaurav Sharma, Don Mundis, Stephen Alberon, Daryl Myshak, Stu Pollock, Fred, and Andrew Swanson, Fredis 104, Sky Luke, Paul Ronovich, Kevin Burden, Noodles Jack, Brian Carrington, Matt Bass, Sean and Nina Lamb, John McKee, Greg Riley, Mark Hassenflow, G. Tellier, Steve Leisman, a.k.a. Zima, Ken Hayes, Howard Tan, Christopher Rappin, Dana Pearson, Richard, Brendan Minish, Johnny Gridley, Kevin Railsback, Rob Me Day, Flying Out, Christopher Dreyer, Artyom, Greg Briggs, John Atwood, Hey Arizona, Support Aaron Lieberman for Governor, Rudy Garcia, Jay Wilkinson, Rodney Lewis, Paul, Mallory, Sutter, Philip, Shane, Kurt Larson, Craig Landon, Mountain Sloth, Jim Drapeau, Sarah Chavez, Sue Doster, Jason Olds, Dave Neighbor, Eric Knapp, E.O., Kevin Parachan, Aaron Luthen, Steve DeBell, Bob Calder, Marjorie, Paul Disney, David Silmerly, Patrick Pecoraro, Tony Steele, Ulysses Adkins, and Jason Roberts. I made it. Only stumbling a few times. Thank you, everyone, for all of your support on Patreon. And if you would like to support us on Patreon, you can find more information at TWIS.org. On next week's show,
we'll be back on Wednesday at 8 p.m., broadcasting live from our YouTube and Facebook channels. And we'll also be at youtube.com/thisweekinscience.
Yeah. You want to listen to us as a podcast? Just search for This Week in Science wherever podcasts are found. If you enjoyed the show, get your friends to subscribe too.
For more information on anything you've heard here today, show notes and links to stories will be available on our website. That's at www.twis.org. You can also sign up for our newsletter or buy one of the few remaining calendars that are still in Kiki's possession. It's not February yet.
No! You can contact us directly. Email me, Kirsten at thisweekinscience.com, Justin at twistminion@gmail.com, or Blair at blairbaz@twis.org. Just put TWIS, TWIS, in the subject line so that your email doesn't get spam filtered into a biodome and integrated into a regenerated frog leg.
Yeah, you don't want that. You can also ping us at the Twitter.
At the Twitter.
Where we are at TWIS Science, at Dr. Kiki, at Jacksonfly, and at Blair's Menagerie. We love your feedback. If there's a topic you would like us to cover or address, or a suggestion for an interview, please let us know.
And we're going to be back here again next week. And we hope that you'll join us again for more great science news.
And if you've learned anything at all from the show tonight, remember,
it's all in your head.
This Week in Science. This Week in Science. This Week in Science. This Week in Science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth. And I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science.